Introducing the BeNano series, an advanced analyzer that combines 3 novel light scattering technologies for precise nanoparticle analysis. Dynamic light scattering for particle size measurement, electrophoretic light scattering for zeta potential measurement, and static light scattering for molecular weight measurement. Particle Brownian motion causes the intensity fluctuation of the scattered light. DLS measures the fluctuation and, through analyzing its correlation as a function of time, calculates the particle size and size distribution. The more advanced backscattering DLS technology adopted by BeNano enables the detection of the scattering light at 173 degrees with a much larger scattering volume. Combined with the movable measurement position, BeNano has much higher sensitivity and high turbidity sample measurement capacity. By adding tracer particles with known size to the sample, the DLS micro-rheology technique can further offer rheological information about solutions. ELS measures the frequency shift of the scattered light due to the optical Doppler effect caused by particles experiencing electrophoretic movement in an electric field, and offers zeta potential and stability information of samples. BeNano adopts the most accurate ELS signal analytical algorithm, phase analysis light scattering, known as PALS technology. It can effectively extract useful signals even for samples with weak electrophoretic mobilities, either with a high salinity environment or close to the isoelectric point. The BAT1 autotitrator is designed to be used with BeNano, performing automatic titration operation and offering a pH-dependent zeta potential plot. SLS measures the molecular weight by detecting the scattered light intensity of a series of sample solutions with different concentrations. By Debier plot, the average molecular weight and the second virial coefficient can be calculated. For thermal sensitive samples, a temperature trend can be performed easily with a programmed SOP. The BeNano software can also detect the temperature transition point of the size results, which is the aggregation temperature for protein samples. Besides multiple powerful functions, the research-level software also provides a user-friendly interface and different types of reports. To ensure data security, it is in compliance with FDA 21 CFR Part 11, allowing permission settings and audit trail. BeNano, as an ideal particle size and zeta potential analyzer, has been widely used for various areas, including pharmaceuticals, proteins and polypeptides, household chemicals, food and beverages, paints, inks and coatings, nanomaterials, and abrasives. As a powerful light scattering device, BeNano helps you exploring the secret of microcosm. BeNano series. Be the nanoparticle expert you need.